You may have heard late last year that Denmark decided to cull its entire population of mink. Here in America, a mink in Utah was the first wild animal found to have the virus. So what is it with the mink? Well, it turns out that mink and related animals have respiratory systems that operate similarly to humans. That's why about 10 years ago, researchers at Erasmus Medical Center in the Netherlands embarked on a series of experiments involving ferrets. They were trying to see if a virus that starts off without the ability to be transmitted through the air can eventually gain that ability. The answer was yes. They discovered that by injecting an influenza virus from one ferret to another until the virus mutated enough to be transmitted through the air by aerosol droplets. This led to the creation of an entirely new type of scientific research made possible by advances in gene technology or genomics. It was called gain-of-function virus research. The idea was to replicate in a lab what had been done with those ferrets, to take a virus and then manipulate its genetic code to make it stronger and more powerful. The virus would gain new functions, hence the name. Gain-of-function virus research, for some, showed huge promise. If we could engineer the most contagious, deadly viruses ourselves in lab conditions, we could learn about potential treatments and vaccines without the calamity of an actual outbreak. The driving principle of gain-of-function research was a noble one. Know your enemy so you can better prepare to fight it. Over the years, gain-of-function virus research has been carried out all over the world with funding from our own National Institute of Allergy and Infectious Disease. And one of the leading laboratories in gain-of-function research was based in China. It's a place the world has come to know well, the Wuhan Institute of Virology. But as well as having great potential to help mankind, gain-of-function research could also hurt us. Imagine if a deadly virus engineered in the lab to be incredibly contagious somehow escaped from the lab. It could wreak havoc precisely because it was designed to do that, not by nature, but by us. For this reason, gain-of-function research has always been highly controversial. In fact, in 2014, the Obama administration went so far as to place a complete moratorium on this kind of research, quote, following recent biosafety incidents at federal research facilities. Here's what they said at the time. The funding pause will apply to gain-of-function research projects that may be reasonably anticipated to confer attributes to influenza, MERS, or SARS viruses, such that the virus would have enhanced pathogenicity and or transmissibility in mammals via the respiratory route. Now, despite the controversy surrounding this type of research, it always had one steadfast champion, a leading figure in the world of infectious diseases, Anthony Fauci. His institute funded that first ferret study. At the time, he co-wrote an op-ed in the Washington Post describing this research as, quote, a risk worth taking. Important information and insights can come from generating a potentially dangerous virus in the laboratory. And shortly before the Obama administration's ban on gain-of-function research in 2014, Fauci commissioned this. It was a project to assess the risk of new coronaviruses emerging from wild animals like bats. One of its stated aims was to see what viruses can infect both animals and humans. Now pay close attention to these words right at the end of the project summary. Predictive models of host range, i.e. emergence potential, will be tested experimentally using reverse genetics, pseudovirus and receptor binding assays, and virus infection experiments across a range of cell cultures from different species and humanized mice. That is a description of viral gain-of-function research, the exact type of research that the Obama administration stopped. But Fauci didn't stop it. He continued on by subcontracting it to this group, EcoHealth Alliance, based in New York. It's run by a big name in the field of virus research, Peter Daszak. He's a zoologist who specializes in wildlife diseases. His claim to fame, and it's a big one, is that he was the person who first discovered the bat origin of the SARS pandemic just over a decade ago. Daszak established that the virus came from bats via a civet cat in a Chinese market. Ever since then, Daszak developed a reputation as the Batman. He and his team would literally go to remote jungle caves to collect bat samples for scientific research. The project that Fauci commissioned gave EcoHealth Alliance more than $3 million over the course of 6 years, despite the Obama administration's ban. And here we come to the crucial step, which has never before been reported. Peter Daszak himself subcontracted a key piece of the Fauci project, the gain-of-function piece. He gave that to a well-known center of expertise in gain-of-function virus research, which specialized in bat coronaviruses. Yep, he gave it to the Wuhan Institute of Virology. The question is, did they give it to us? Let's see what they did with the Fauci money. In the project's results page, there are 13 papers co-authored by Shi Zhengli, who is head of the Wuhan Institute's infectious disease unit. One of the papers was published in 2017, roughly halfway through the project. Peter Daszak is listed as a co-author. It's a kind of progress report. Inside the funding section of that paper is the ID of the Fauci Commission project. That's the money trail. Until now, the only reporting on Fauci's role in all this has been on his funding of the Wuhan lab in general. But tonight we can go further. Thanks to this paper, we can see the specific activity that Fauci funded. And it's terrifying. So terrifying, in fact, that after we reached out to the NIH and Peter Daszak's group for comment, this paper was mysteriously taken offline on Friday for a while. But we downloaded it weeks ago. Here are the details. We're putting the actual scientific language on screen now, but this is what it means in plain English. They collected samples of bat feces from a cave near Kunming in Yunnan province, China. They found many novel bat coronaviruses in the feces. They analyzed them and sequenced their genetic information. They then built various chimeras, genetically engineered new viruses, man-made, in the lab. They infected human cells with them in the lab. And they then showed that their man-made viruses could replicate as a functional virus. Here's the most important part. The genetic changes they made in the lab unlocked a highly specific doorway into the human body. The virus that causes COVID-19 uses that same exact doorway. To be clear, none of the genetically engineered viruses created back in 2017 and reported in this paper were the exact ones we're dealing with now. But what they confirm is that it was possible to make the COVID-19 virus using the techniques developed in the Fauci project. And that project continued for 3 more years. 3 more years of experimentation, 3 more years of man-made mutation. Now let's look at the actual COVID-19 virus. How does it compare to the ones we know were made earlier in the Wuhan lab? The actual pandemic virus was found to stick to human cells 10 to 20 times tighter than the original SARS virus, which is why it's much more infectious. Scientists would expect increases in infectiousness to happen through cycles of mutation, but with this virus, it was there from the start. In a paper published in February 2020, Shi Zhengli confirms that the backbone of the pandemic virus matches one from the Wuhan Institute's library of collected samples. Their 2017 paper describes how they swapped parts of coronavirus found in other animals into bat coronavirus strains in order to make more infectious man-made versions. And sure enough, the pandemic virus looks like a combination of 2 very different bat viruses mixed with parts of a virus from a completely different species. Could something like that occur in nature? That's what Peter Daszak is saying, quote, maybe a farmer got infected or a farmer's animals and they were shipped into their markets. If a person brings it in or an animal, that virus will spread. And it looks like that's what happened here. If we're to believe that natural origin story, here are the coincidences that would have had to happen. Multiple different species of bats in South China would have to infect each other and some other unknown animal. Then either these animals or an infected human would need to have traveled 1,000 miles without infecting anyone until they got to Wuhan. In its first incarnation, before any mutations, it would have been 10 to 20 times more infectious than any previously observed virus. And most incredibly of all, the infected animal or human would have somehow chosen to make their 1,000-mile trek to the only place in all of China with a lab that had the world's most extensive collection of the viruses it was already infected with, and the one place in the entire world that carried out genetic research which matched